Let's look in a little more detail at HIV, specifically the Pol region. And by Pol region, I mean we're not going to cover the protease, we're going to cover the reverse transcriptase part and the integrase that comes right after it. I want to show you a multiple sequence alignment and some structures. Get it in your head a different way, kind of what's going on, how these little machines work. And we'll start off our background music. This is going to be the Sounds of HIV from Alexandra Payak. She actually turned the genome into music long before generative AI was a thing. So it's nice, peaceful. I believe the part we're listening to is the 5' UTR, basically the LTR section. So there you go. All right, let's talk HIV. So look on the screen right above me. You can see that spinning molecule there. That is the reverse transcriptase. And remember, reverse transcriptase has two parts. So our greenish part, or the bigger part, is going to be called P66. That's the active part of the reverse transcriptase. The inactive part is the orange part that's right here. I'm sort of circling it. That's going to be the P51. The difference between these two is that the RNA's H domain is cut off to make the P51, and it just sort of folds up. It sort of crunches down. Now, if you're looking at the structure and you want to see, all right, where are all these parts that we've heard about in class? If we circle this little bit right there, that's the palm domain. So those little beta strands are shown as ribbons, and the palm is made of beta strands. So that's where the actual, like, catalytic active site would be located. This sort of blob at the bottom, that's going to be the fingers, and this little amalgam of 3 alpha helices, that is the thumb. And then you got the rest of the enzyme sitting back here. It sort of holds those parts in place. And the RNA's H domain is this bit up here, only on the green molecule, and that's the part that's missing from the orange molecule. All right, so that's the structure. Now let's look at a multiple sequence alignment. So multiple sequence alignments I like because they show you what's important in the molecules, because stuff that's important will be conserved usually. Like, it's got a functional job to do, and there probably aren't many ways to do that job, and so there aren't many ways that it can change. In something as hyper evolved as HIV, you will see specific points where the virus has been unable to come up with another solution, despite having nearly infinite attempts at evolving around these. And so you know that these are important. All right, so what we're looking at here, this is called clusteral omega coloring, and I like it because it's only going to color in amino acids. So we've got our single letter amino acid codes. And if you don't know your single letter amino acid codes yet, now's a good time to pause the video, go learn them, then come back, and it'll make more sense the next time. You'll still get something out of it now, I figure, even if you don't. All right, so we're starting off, and the clusteral coloring is only going to show up colors, and the color's going to tell you what sort of amino acid it is, and it's only going to be colored if that amino acid is conserved, meaning it appears a certain percentage of time, and it's got its own rules for each one, and yeah, that's fine. So if it's colored, figure it's more important than if it's not colored. All right, so we're starting off. This is the N-terminal domain, so this is going to be what folds up into the fingers and parts of the palm sort of support structure, kind of around the back of it. What you've got here are a couple important motifs. So this right here, this LKKK, the top one is actually HIV-1. The others are different retroviruses. So it's retroviridase. We're looking very broad brush. What's conserved in all retroviruses? You know, how does the general machine work? Not how does specifically, you know, one version of it work. All right. So we're not showing GAG, and we're going to show Pol, but we're not even going to show the end of the integrase, and we're not going to show the protease. Yeah, this is only the most conserved parts, and those are the reverse transcriptase and integrase. All right. So you come up here, this LKKK, that is the main spot where non-nucleoside reverse transcriptase inhibitors bind. It's like a little hinge. See, it's very positive, at least in HIV, but you can see the positive is not conserved. because conserved positive amino acids show up as red columns. So like here's some K's that are sometimes R, sometimes K, and yeah, that's how you can tell. Conserved acids, acidic residues, like with a negative charge, are going to show up as purple columns. And you'll have the occasional not conserved residue that's like this E would be purple if it was part of a whole column, but you can see clearly, no, there's a bunch of different letters there. That position is not conserved, which means it's probably in some sort of loop, or it's structural. It's not doing a major part of the chemistry that makes HIV reverse transcriptase and integrase work. All right, so right after the NNRTI binding spot, which is right there, you're going to have your major nucleotide binding spot. So this is where the nucleotide that's going to be added on to the end of the strand that it's extending, like the end of whatever the primer is, whether that's your tRNA or whether we're just the next nucleotide in the chain, and DNA or RNA, or rather, yeah, DNA, because we're going to read DNA or RNA, but we're going to write DNA, of course, because it's a reverse transcriptase. It's going to be this little motif here. So this D that's always conserved and this other D that is usually conserved, those are going to sort of frame the binding pocket for the nucleotide in 3 dimensions. Yeah, so there we go. And so if you have inhibitors that bind to the nucleotide binding pocket, which many of them do, like all the nucleoside analogs, then that's where they're going to bind on the protein. Now, when looking at an alignment, you can see that a protein is just a big long spaghetti squiggle that folds up into a shape. And if it makes little helix shapes, we color that as like a little ancient phone cord, like a little coil. And if it's sort of stretched out, then we're going to call that a beta strand. And you'll see those like over here on the side or down here in the active site. All right, so just because 2 things are next to each other in the alignment doesn't mean they are necessarily part of the same feature or particularly close in the protein. Likewise, things that are far away can get folded up so that they are right together. Yeah, that's proteins, yeah. It's just a different way of thinking about things, but once you get a hang of it, it's not too bad. Okay. Moving on, let's find the active site. Now the active site is going to be here, and it's either MDD or VDD sometimes, and you can see that's in a pocket that's pretty conserved. You've got a lot of these light blue residues, which are hydrophobic, so sort of a hydrophobic pocket with 2 very acidic residues, kind of dead center in it, and also this Y over here, which is a big aromatic residue. That one is going to also participate in binding. Now these Ds are going to help to coordinate magnesium ions, and it is a classic 2 magnesium ion mechanism. So they're going to use these 2 ions to actually do the chemistry, to take the nucleotide from the nucleotide binding spot and add it onto the chain. The rest of this molecule is going to be holding on to the template strand and holding on to the primer and just supporting all these parts so that they end up in the right 3-dimensional context. All right, scrolling forward, we get to this chunk here from about 230 to about 275 in the alignment. That is the thumb. So this is the bit that's going to fold into 3 alpha helices like that, and that is the thumb. And that's the bit that just rotated around. I'm sort of circling it here in green. And so that's going to be a mobile part. You can see it's got a little flexible hinge down here, and so it can sort of close that active site, which we think it does to some extent during catalysis. All right, let's scroll on. So now you got the rest of the enzyme. This is the part that's going to sit around the back here. And then you get over here, and boom, once we get to this spot right here, a little around 485, now we're in the RNA's H domain. So the cleavage that makes P66 different from P51 has to happen in this vicinity. And you can see the cleavage site is not particularly conserved, so it's always going to be cleaved by something, but it's not always the same something in the same way, and that's fine. Cells have lots of proteases. All you have to do is attract one, and it doesn't really matter. It can work with almost anyone. Okay. Now, this is a nuclease. So it's an RNase H. It's a thing that can break the RNA side of an RNA DNA duplex. And the way it's going to do that is it has a motif. It's called a dead box motif. Now that's dead spelled D-E-D-D. Yeah, not the correct way, but the correct way for this enzyme. And those are the important amino acids that are going to do the catalysis. And so here's the first D. You can see that's conserved in every single one. Here's the E, here's the D, and you got to scroll right to the end. There's your other D. So those four end up folding up in 3 dimensions, and they're right next to each other, and those are the ones that are going to actually do the catalytic activity of the RNase H. So that's cool, right? All right. So after that, now we're out of the reverse transcriptase, and now we're beyond. The molecule is up here. Yeah, don't even look at that ever again. I don't have the integrase loaded up, otherwise I would switch to that. So here you have a linker. And the linker, as you can see, is not very well conserved, which just means you have a reverse transcriptase and you have an integrase, and like 2 train cars, you have something that's connecting them, and that something will be kind of flexible. So everywhere you see like a G or a P, those are going to be spots where they are interrupting any structure that could have formed. They're just going to be floppy bits of a length. All you have to do is separate these 2 cars. You just can't have them joined together because they bump into each other. So yeah, different size connectors. Okay, and then after the connectors, we are into the integrase. So starting out, here's domain 1 of the integrase. And domain 1, you'll remember, is a zinc binding domain. Now how do you bind zinc? Well, you have 4 histidines, that's amino acid H, or cysteines, that's amino acid C. Usually there are more cysteines than histidines, but theoretically you can do it with 4 of any of those. And they are going to pinch it in a tetrahedral grip, which means like take your 2 thumbs, rotate one like that, and just imagine they're all touching the same thing in the center. Boom. There you go. That's your tetrahedral grip. And so you can find those here. Look, that histidine is completely conserved. That's the H. Here's another one. So that's number 2. And can you see 2 cysteines? They come up in sort of a salmon pink color. Yeah, right there, right? Boom, boom. So in 3 dimensions, those 2 cysteines and those 2 histidines are going to fold up just like so, and they're going to pinch a little zinc atom between them. A nice little divalent cation, which is going to help us stick down to DNA. And it'll help the rest of the protein to fold around this little module. Okay. Moving on, so that's domain 1. Now we're going to have domain 2. And domain 2 is going to have a catalytic site. Now it's not a dead box, but it is a DDE box. So it's kind of the same thing. And the DD and E aren't going to hold on to magnesium as they did for the polymerase. They're going to hold on to manganese. And so it's this D right here that you can see is always conserved, this D that's always conserved, and this E right near the end that's always conserved. And that's the end of the alignment. Now, if you're a student of the HIV integrase, which you should be by now, you'll say, hey, wait a minute. Yeah, there's 3 domains in the integrase. The 3rd domain is a generalized DNA binding domain. To which I'd say, well done. Yeah, you got it. You know your integrase. Good job. Yeah. And that part is not very well conserved, and so they cut it out of the alignment. I pulled this alignment down from the ICTV website. But if it's generalized DNA binding, you would expect to see some conserved basic amino acids like Ks and Rs. Those are going to help just get a generalized grip on DNA. And so, there you go. That is the Pol region, sort of in structure above and in amino acid form over here in our alignment, so you can kind of see what's going on. Hope that helps and gives you a little more that you can talk about on a molecular level. Thanks very much.